All right, next we're going to hear from Madison Woodburn. Madison is actually the younger sister of another Eckerd student. So now we have a generational thing going on. Blade Woodburn. Some of you might remember him. Madison is actually his sister. She is a molecular biology major. She's a rising junior, and she comes from Chandlersville, Ohio, and she spent the summer in Sue Cotman's lab. So Madison.
Hello. As Sue mentioned, my name is Madison Woodburn, and I go to Eckerd College. And this summer, I work in the Cotman lab. And I will be talking to you about the development of a drug sensitivity assay and marine cell model for CLN3 disease. Let's see. Now, a slight overview of this presentation. I will talk to you all about what neuronal ceroid lipofuscinosis is, and a specific genetic subtype called juvenile neuronal ceroid lipofuscinosis. Then I will get into the development of a Dapsagargan sensitivity assay, and then I will get into the discoveries and further exploration, and lastly, I will talk about my acknowledgments. Neuronal ceroid lipofuscinosis is also known as Batten disease, and it's a group of neurodegenerative disorders defined by shared clinical and pathological characteristics, including visual failure, dementia, behavioral changes, motor difficulties, seizures, and lysosomal storage buildup. And there are 13 different genetic subtypes, and the Cotman lab mainly focuses on CLN3 disease, also known as juvenile neuronal ceroid lipofuscinosis. And over time, the Cotman lab has created a series of cell lines pertaining to specific phenotypes discovered in patients that have the exact CLN3 mutation. And notable phenotypes include lysosomal phenotypes, mitochondrial morphology, autophagy, and there's a way to measure abnormal autophagosome accumulation with a biomarker called GFP-LC3, and it marks autophagosome vesicles, as shown here. And as you can see in mutant GFP cells, you can see increased GFP-LC3 accumulation in mutant type cells compared to wild type cells. And it has been recently discovered that in addition of Dapsagargan, which is a circuit inhibitor, there is a significant rise in GFP-LC3 accumulation in mutant type cells compared to wild type cells. And it has been quantitatively represented in the graph to the right. And preliminary data has led us to believe that this might be due to calcium influx, ER stress, autophagy inhibition, or maybe that circuit interacts with CLN3 in some way. So my project this summer was to understand the mechanism of action of Dapsagargan and develop a platform for testing therapeutic compounds. So when developing this Dapsagargan sensitivity assay, we created a hypothesis, which was compounds that rescue other CLN3 phenotypes will be able to revert this Dapsagargan sensitivity. And various compounds utilized were verapamil, which is a calcium channel blocker, and it has been previously shown to rescue lysosomal phenotypes and GFP-LC3 accumulation. And a novel compound used in the Cotman lab was CDN1163, which acts exactly opposite of Dapsagargan, which is a circuit inhibitor. CDN1163 is a circuit activator. And this is a general template of our Dapsagargan sensitivity assay, and it represents our goal. And our goal is to develop a Dapsagargan drug sensitivity assay and test if various compounds such as verapamil and CDN1163 will be able to revert Dapsagargan-induced GFP-LC3 punctate formation. And when creating this drug sensitivity assay, different orders and incubation times were tested for assay development and with various studies designs. So our first treatment was our rescue treatment. And what we did was we exposed mutant and wild type cells to Dapsagargan for 24 hours and incubated them. Then we added the test compound for 24 hours. Our next one was a co-incubation type treatment, where we added both test compound and Dapsagargan to mutant and wild type cells and incubated them for 24 hours. And the results of these two types of treatments were that the cells did not like these two types of treatments at all, as all cells died. So we went to a prevention treatment where we added the test compound to both mutant and wild type cells, incubated them for 24 hours, and then we added Dapsagargan and exposed them for 24 hours. Now, the results of this treatment was that there were some cells that expressed decreased GFP-LC3 accumulation, but some cells did not survive. So this led us to believe that maybe Dapsagargan exposure for 24 hours was maybe too long. So we decided to fine-tune this prevention treatment by keeping the same protocol by exposing the test compound to both mutant and wild type cells for 24 hours, but then doing a 4 and 8 hour treatment. And the result of this was that the 8-hour Dapsagargan treatment post-test compound was the best study design to utilize in our assay. Now, before getting into the actual 8-hour prevention assay treatment, we developed a control treatment to test various concentrations of the compounds used so we can compare it to the 8-hour prevention assay. And as it has been previously shown, when you add Dapsagargan to both wild type and mutant cells, there's increased GFP-LC3 puncta per cell, especially in the mutant type cells. And when you add Dapsagargan to mutant cells, there is decreased GFP-LC3 puncta per cell, as previously noted. And intriguingly, when you add CDN1163 to mutant type cells, there is decreased GFP-LC3 puncta per cell. And this was very interesting to see because this compound has not been used in the Kottman lab before to measure autophagy. Now, getting into our actual 8-hour prevention assay, it showed decreased GFP-LC3 accumulation in mutant cells. And using our mutant cell with Dapsagargan as comparison, we can see that in addition of verapamil in various concentrations, we can definitely see a decreased level of GFP-LC3 puncta per cell. And what was statistically significant was the Dapsagargan plus verapamil at 10 micromolar and Dapsagargan plus verapamil at 16 micromolar. And then when CDN1163 was added in various concentrations, there was a slight decrease in CDN1163 at 0.1 micromolar and a statistically significant decrease in GFP-LC3 puncta per cell in 1 micromolar. So our conclusions of this project was that our goal was succeeded. We successfully developed a Dapsagargan sensitivity assay. And with verapamil, it showed a dose response in preventing Dapsagargan sensitivity. And with CDN1163, a circuit activator, it also was able to prevent Dapsagargan sensitivity. And future experiments entail using this assay to further study certain mechanisms related to Dapsagargan sensitivity. And the Kottman lab is currently performing an autophagy flux assay. And then autophagy induction in CDN1163 is being further explored. In the future outlook of this drug sensitivity assay, they will be able to use this assay to test certain drugs for therapeutic benefits. Then here are my acknowledgments. Thank you to everyone. You guys contributed to my career aspirations immensely this summer, and you guys showed me how much I love research. Thank you.
Thanks, Madison. Any questions for Madison? It was really great, Madison. It was awesome that you were able to develop that assay that will be used. A quick question, the drug, the CDN 1163 drug, was that something that was screened for and identified in the Kottman lab, or do you know where that came from?
No, so what we were trying to do was when we were creating our hypothesis, we were trying to find different compounds that we can use. And I came across CDN1163, which acted exactly opposite of Dapsagargan. So we decided to just say, okay, let's try this and see how it works out. And it turned out to work out really well.
Great, great. Thanks so much. Any other questions? Okay, thanks, Madison.